So today our topic is meningitis. So meningitis is also asked as a clinical scenario based questions in the long answer type questions, and they do not give you directly about meningitis, rather they give you a clinical scenario and you have to diagnose the case based on that clinical scenario and then answer the other questions. So whenever the clinical scenario is like a patient comes to the OPD or to the emergency with fever, vomiting, intense headache, or most importantly, the nuchal rigidity, that is, stiff neck. So, if any patient comes to the emergency or the OPD with this nuchal rigidity, mostly the patient come to the emergency in case of meningitis. So, if the patient comes with the presentation of the nuchal rigidity, stiff neck, then you have to diagnose the case as the as a case of the meningitis. Sometimes some other informations are also given like the Karnik sign, Brudzinski sign as well. These signs are positive in case of meningitis. So if these tests are also given, then it is also better for you to say or diagnose the case as the meningitis. But if there is nuchal rigidity, generally it is meningitis. So if there is nuchal rigidity, you can safely diagnose the case as meningitis. These are extra points. If given, then you can surely say that it is meningitis. If not given, then don't worry, it will be meningitis if there is stiff neck. Now coming to the causative agents. So again, here we classify the causative agents as bacterial causative agents, viral causative agents, and the fungal causative agents. Now, the bacterial causative agents are divided on the basis of their frequency of causing meningitis in different age groups. Like overall, if we talk about overall, then these five bacteria are most common to cause the meningitis. These are Streptococcus pneumoniae, Meningococcus, Streptococcus agalacti, Listeria, Haemophilus influenza. Okay, so these are the five bacteria which are overall most common to cause the meningitis. In the case of neonates, the Streptococcus agalacti is more common to cause the meningitis, while Listeria monocytogenes and E. coli are also common to cause the meningitis in neonates. And in case of elderly, the Streptococcus agalacti and Listeria monocytogenes are more common. Especially this Listeria monocytogenes are found in some some food materials like pickles and all. And this why in case of neonates Streptococcus agalacti is more commonly causing this meningitis, because the mother's genital tract in the mother's genital tract, these bacteria are present as commensals. So while normal vaginal delivery, while the passage of the baby through that vagina during at the time of delivery, they get infected with this Streptococcus agalacti, and that's why the Streptococcus agalacti is the most common cause of meningitis in case of neonates. Now, if we talk about overall, so in overall, we have five bacteria which are most commonly causing meningitis. You can remember them as SMS Lee hai, SMS Lee hai, or you can say is it as SMS key hai. So S for Streptococcus pneumoniae, M for Meningococcus, Streptococcus agalacti. Key is similar to Lee, so Listeria, and hai means Haemophilus influenza. So that is a trick to remember the five bacteria if you want those. So now coming to the viral causative agents. So viral causative agents, there are many viral causative agents, out of which we have the Coxsackie virus, the ecovirus, also called as the orphan virus, the herpes virus HSV, the varicella zoster virus, and the mumps meningitis. So mumps virus is also found to cause this viral meningitis. So these are the five viruses which are causing meningitis. Now coming to the fungal causes. So in fungus, the most common fungus which is causing this meningitis is the Cryptococcus neoformans. Is the Cryptococcus neoformans. Remembering these all name is very important. This is sometimes asked as a short note question and carries about 4 to 5 marks also. So this information is very important. This information is very important from the exam point of view. Now coming to the lab diagnosis part, which is also a very important sections of meningitis. So coming to the lab diagnosis part, we have first the in the lab diagnosis of any condition, first we have to collect the specimen. Now we know that in case of meningitis, the infection is to the meninges, which are the covering layers of the brain parenchyma. So yes, the what will be the sample for us? So sample will be, or the specimen of choice will be the CSF, because the infection is occurring in the meninges, and close to the meninges is the CSF. So the changes will be found in the CSF. That's why CSF is the ideal specimen. Now the question comes is, how will we connect the specimen? How will we collect the CSF? So see here how we are going to collect the specimen. See, CSF collection. So CSF is collected by lumbar puncture. So we will first uh, make the area aseptic. The generally we take this CSF from the L3 L4 vertebra level, and so first that area is made aseptic, and then by the method of lumbar puncture we collect the specimen, or the CSF, which is the CSF. So specimen, after the as we are collecting the specimen, we will collect it in three separate containers which should be sterile. Now why three? Because one container will be sent to the microbiology lab, one will be sent to the pathology lab, and the third one will be sent to the biochemistry lab. So that's why we collect in three different containers. Now other than CSF, blood may also be collected. Sometimes blood is helpful, not in confirmation, but in supporting the present evidence. So blood sometimes helps, so that's why blood is also sometimes taken as the specimen. Now coming to the specimen transport, how are we going to transport the specimen once it is collected in three separate containers? So the specimen should never be refrigerated. Mind this, that the CSF specimen should never be refrigerated. Why? Because if there will be H influenza meningitis, that H influenza is cold sensitive, and that H influenza will die. So that's why we never refrigerate the CSF specimen, just because of the H influenza. Now if the delay is expected, because if the lab is closed or the lab is situated far away, then we can incubate it, but never refrigerate it. We never refrigerate it, but we can incubate it at 37 degree centigrade. So these 2 points should be kept in mind. It should never be, refrigerated. And the point number 2 is that we can incubate it at 37 degree centigrade. Now the third point is the cytological and biochemical analysis. So as we have sent the three containers to the microbiology lab, pathology lab, and the biochemistry lab, so we will get the reports from all the three labs. After getting the report from all the three labs, what are the cytological and biochemical things that we see in the report? So after getting that report, we will analyze the cytological and biochemical reports. In that analysis, we have CSF pressure, the leukocyte count, predominant cell, glucose, and total protein. So CSF pressure If it is a pyogenic meningitis due to any bacteria, then it will be raised. If it is a tubercular meningitis, that is also a bacteria, but it is given a separate category because it has some different characteristics for its identification. Okay, like in tubercular meningitis, there is a cobweb formation in the CSF when it is kept for some duration. Okay, so that is identification point for the tubercular meningitis. Leave that. Now we were talking about the CSF pressure. So in this tubercular meningitis also, the CSF pressure is moderately raised, but in viral meningitis it is either raised or it is normal. The leukocyte count is raised in case of bacterial meningitis, pyogenic meningitis. In tubercular meningitis also it is raised. In viral meningitis also it is raised, but the predominant cell varies. Like in pyogenic meningitis, the predominant cell in the CSF will be the neutrophil, but in tubercular or the viral meningitis, it will be a lymphocytes. So lymphocytes will be in excess in the CSF in case of tubercular and the viral meningitis. But in case of pyogenic meningitis, neutrophils will be in excess in the CSF. Now glucose is falling. Why glucose is falling? Because bacteria is utilizing the glucose present in the CSF. So that's why glucose is very much falling in case of pyogenic meningitis. And tubercular bacteria also utilizes that glucose. So also in that case also the glucose level falls. But viruses do not utilize glucose. So glucose remains normal. This is a very important point for identification whether it is a bacterial meningitis or a viral meningitis. In viral meningitis, the glucose level in the CSF remains normal, but in case of pyogenic meningitis, the is glucose level fall to a very low level. Total protein increases in case of pyogenic meningitis because the bacteria synthesize some of the proteins. In tubercular meningitis also it increases, and in viral meningitis it is either raised or it is normal. Now there is we have seen that the predominant cell in pyogenic meningitis is n- neutrophil, but in case of meningitis due to listeria, remember this. Listeria was a common cause of meningitis in elderly people. So, in case of meningitis due to listeria, the predominant cell is the lymphocyte. The predominant cell is lymphocyte, and in all other cases of the pyogenic meningitis, the predominant cell is the neutrophils. So, this is an important point about the listerial meningitis, that the lymphocytes are raised in the CSF in case of listerial meningitis. Now, now, now coming to the CSF microscopy after gram staining. So, we will make a smear with the help of that collected CSF, and then we will gram stain the smear, and after gram staining we will examine it under the microscope. So what type of smear do we prepare with the CSF? We prepare a heaped smear. Why? Because the bacterial load in CSF is very low. So several drops of the CSF are placed at same site, one after the other, and heaped smear is prepared. Other than this, the CSF can be centrifuged also and deposited at, and the deposit which is formed at the base can be used to produce the smear. So we have two ways to produce the smear with the CSF. Either we can produce a heaped smear by um putting the CSF drop by drop, one after another on the slide, and letting it to be dried. So we can prepare a heap and thereby a heaped smear, or we can do a centrifuge. We can do a centrifuge of the CSF, and the deposit can be used to produce the smear. After the production of the smear, we will gram stain it. So smear is stained with the gram stain, and then it is examined under the microscope. Why this gram staining is important? Because this gram staining will give you certain clues about the causative bacteria. Like if there is a gram positive lanceolate shaped diplococci which is capsulated, then you can think in terms of streptococcus pneumoniae as a causative agent. If it is a gram negative lens shaped diplococci capsulated, then you can think in terms of Neisseria meningitidis. If it is a gram positive cocci in short chain, then think in terms of Streptococcus agalacti. If there is a gram positive small bacilli, then think in terms of Listeria monocytogenes. If there is pleomorphic gram negative coccobacilli capsulated, then think in terms of Haemophilus influenzae. So this is the clue that we get. This is the clue that we get by gram staining. That's why we do the gram staining with the CSF. After gram staining, we can do culture. So for culturing the CSF, we use enriching media. Why? Because bacterial load in CSF is very low. So a small amount of CSF is inoculated into the blood culture bottles or the BHI broth, and then from so in the enriching media the number of the bacteria increases. Then we use the inoculate with those bacteria in the culture media. What culture media do we use blood agar and the chocolate agar. So after the growth of the bacteria into the enriching media to where we have kept those kept that CSF for increasing the or multiplication of the bacteria, increasing the number of the bacteria. So after the enrichment in the enriching media, then we inoculate the sample in the culture media, that is the blood agar and the chocolate agar. And then we on the culture media we see the colony characteristics. Like we see the colony characteristics like on the blood agar if there is alpha hemolytic Dortmund shaped carom coin colonies, then we can think in terms of streptococcus pneumoniae. If there we see satellitism when staph aureus is streaked on the blood agar, then we can think of Haemophilus influenza. And if there is production of beta hemolytic colonies, then we can think of Streptococcus agalacti and Listeria monocytogenes. So remember, these colony characteristics are helping us in getting a clue about the causative agent. So that's why they are also important as they are helping in the lab diagnosis of the bacteria. Now after the colony characteristic, we gram stain. So we take a colony from the culture media and we make a smear on a clean grease free glass slide, and we stain it with the grams stain, and then the morphology of the bacteria is seen, and that gives a clue about the causative agents, the morphology of the bacteria that we have talked earlier. This gives a clue about the bacteria. Now after this all, what comes next is the identification. So we have to confirm which bacteria it is. So for confirmation we can use the automated identification systems like Malditov and Vitek, or we can use the conventional biochemical tests which are not done nowadays. These conventional biochemical tests are not very much commonly done nowadays. We generally use the Malditov and the Vitek automated system for because of the increasing loads of the samples in the laboratory, and also it is easy to do it in the automated identification systems. Then we have the molecular methods. molecular methods. Why? Because they are more sensitive and more specific, because we uh, identify the bacteria at the genetic level by the molecular methods. That's why the molecular methods are much more sensitive and specific. So in molecular methods we have PCR, which detects a specific gene in the bacteria. We have biofire film array, which is automated nested multiplex PCR commercially available. So that can be also used for the determination of the determining which bacteria it is. Then we have antimicrobial susceptibility testing. This is most ignored part by the students while writing the answers in microbiology. So you have to add this point at last. These points can be these points sorry these points can be added in any lab diagnosis, molecular methods and antimicrobial susceptibility test. So these points can be added in lab diagnosis of any bacteria or any case. So make sure to add these 2 points, molecular diagnosis and antimicrobial susceptibility testing in all cases. So what do we do in antimicrobial susceptibility testing? The resistance against the antibiotics is searched in the bacteria. Like we search for the antibiotics to which the bacteria are susceptible, so that we can use those antibiotics and kill the bacteria. So this is done by disk diffusion method on the Muller-Hinton agar. This helps in accurate treatment of the condition. Okay, so that's the importance of the antimicrobial susceptibility test. This is all about the lab diagnosis of meningitis. Now, one MCQ sometimes asks, that's why I have added it here, that what is the complication of the meningococcal septicemia, or what is the danger of the meningococcal septicemia. So remember, it is sometimes it is asked as MCQ. So it is Waterhouse Frieder Fried Friedrichsen syndrome. Waterhouse Friedrichsen syndrome, in which there is multi-organ failure, shock, disseminated intravascular coagulation, and the petechial rhesus. This is all about the lab diagnosis of meningitis.